co-workers, Amy Smagula and Josh Bonpen. Amy has been my partner in crime for a very long time with the spiny water flea monitoring. She's basically my other half when it comes to doing this monitoring. So it would not have been possible without all her efforts. And Josh is a fairly new employee with DES. He's been with us for a couple years. He moved into my former position as a biologist, and he has brought a lot of insight and expertise, and he assisted with some of the analysis and plotting of the data. He's a really smart guy. So I would not have had this presentation to give as nicely as it is set up if it weren't for these two. Okay, so before I delve specifically into spiny water flea, I want to talk about invasive aquatic species in general. So with invasive species, the moment we're currently in is really unprecedented. We have species being introduced into novel habitats that they didn't evolve in across continents all around the world at a frequency that's never been seen before in the history of the planet. So when it comes to these aquatic invasive species, they're having a huge impact. One study from 2021 conservatively estimated that the global impact of these aquatic invasive species was cumulatively $345 billion. That was several years ago now. These invasive species can have direct impacts on the native species and the ecosystems that they are invading. They can outcompete native species, they can eat them, they can basically just take over. There's also indirect impacts that they can have. They can alter energy flows, they can change native species behavior or change the gene flow. So there's a lot of different ways invasive species are changing and impacting the ecosystems that they are finding themselves in. So when we're talking specifically about spiny water flea, it was first discovered in 1984 in Lake Huron, and it can get up to 15 millimeters long. Now that sounds really tiny, and you can see from the picture that, or you'll see on the next slide, I can see the other slide, so I'm getting ahead of myself. They're not that big to us, but when it comes to the zooplankton world, these are huge predators. They will just get in there and eat all the zooplankton that they come into contact with. And as such, there's a documented decline when they invade certain water bodies that they will decrease native predator abundance, such as the zooplankton Leptodora. They've been found to reduce pressure on phytoplankton sometimes. They can be avoided by young of year fish because of that long spiny tail. So there's a lot of different ways they can impact the ecosystem they've come into. They're native to Europe and Asia, and they prefer large, low nutrient temperate lakes. So New Hampshire has a lot of those. While they're not harmful directly to humans, they do have all these direct and indirect impacts that I was outlining. And also there's a little bit of debate over the correct taxonomy, the correct Latin name for spiny water flea. So you will find it referred to by both Bithotrepes cedarstromi and also Bithotrepes longimanus. A lot of the literature I see out there calls them Bithotrepes longimanus, but there was another paper in 2019 that was specifically a taxidermy paper that called them out very strongly as Bithotrepes cedarstromi. So I have both here for your reference. So spiny water flea in New Hampshire was first documented in Lake Winnipesaukee in September of 2023. It was the first recording of spiny water flea in New Hampshire, and more were found in Lake Winnisquam not long after, within a month after the first recording in Lake Winnipesaukee. They were documented in a third lake, confirmed in Newfound Lake in 2025. And they, at least in Lake Winnipesaukee, they were first confirmed by a fisherman. And we followed up a couple weeks later, and sure enough, there they were. Winnisquam is downstream of Lake Winnipesaukee hydraulically, so we believe that that connection probably allowed spiny water flea to move into Lake Winnisquam. And also because of that hydrologic connection, we do assume they are probably in Lake Opeechee as well, because it's in the middle of Lake Winnipesaukee and Lake Winnisquam, but we have not been able to confirm this yet with a live specimen. So 3 confirmed lakes in New Hampshire, probably at least one more. Was this an unexpected invasion? And I would have to say both no and yes. We actually started monitoring for spiny water flea on Lake Winnipesaukee in 2016. This was driven by my interest in invasive species ecology. I had a background in it before I came to DES, and I was very interested in staying involved in invasive species monitoring and kind of keeping my toe dipped in that world. And since we knew that spiny water flea had been discovered in Lake Champlain in Vermont in 2014, I partnered with Amy Smagula at the time, and at the time she was our exotic species coordinator, to implement a monitoring program. So what we did is we located 9 deep spot locations around Lake Winnipesaukee. And once a year, typically in late summer, but we would always have to schedule around our other field work and obligations. Once a year, we'd make it out to Winnie and do a horizontal plankton tow. We would use 2 different nets. One we called a 80 micron net, which the micron is basically just the size of the net mesh. So 80 microns means it's a very small mesh, and it was intended to target things like phytoplankton and rotifers, which are really small zooplankton. And then I also had a 250 micron net, that much bigger net. The picture that you saw at the beginning of this presentation, that's the 250. And that net was intended to target spiny water flea and more macrozooplankton, that should say macrozooplankton. So 2016 was our pilot study. However, at that time, we were still kind of ironing out our monitoring efforts and our sampling protocols. So because we didn't have everything totally locked down at that point, the data and information I'm going to be showing you tonight is going to focus on 2017 up through 2025. So as I said, although we started monitoring in 2016, we did end up finding spiny water flea in 2023. To be honest, it did feel a little unexpected. You know, we'd been doing this for several years without a sign of it, and it kind of just felt like, you know, hey, we're getting an idea of the plankton community on Winnie. It's good for us to have an eye on that, but you know, that we actually found the invasive species the program was intended to find was a bit of a shock, to be honest. Now, that first year in 2023, we found spiny water flea live specimens at 4 of our 9 locations on Winnipesaukee. 2 locations had one of the long spines, but broken off from a body. So it was just the spine and not the actual whole spiny water flea itself. And there were 3 stations that did not have any spiny water flea at all in that first year. So then the following year, the population had seemed to have spread. Now we were finding it at 8 out of 9 stations in 2024, with just Molten Burrow Bay being the only place where we did not pick up spiny water fleas. And then a little surprising, fewer stations in 2025. That circled station, Alton Bay, down in the south, didn't have any spiny water flea specimens in the samples last year. So 7 of our stations had confirmed individuals. And once again, Molten Burrow Bay did not have any spiny water flea. So before I get into the results of what we've found over the last few years, I just want to tell you a little bit about how we looked at the data and what it's going to look like. So I analyzed these data using a Mann-Kendall trend test. This is just a very simple test that says, is there a trend? Yes or no. When I'm plotting the data, I use box plots. And as an example on the screen, the middle line in the middle of the box is your median line. And then 25% of the data is in the top part of the box and 25% is in the bottom part of the box. So 50% of all the information is contained in the box. Then the whiskers, the more, as you're getting to more outliers as you go along, that's the final 25% in either direction. Now, as I mentioned earlier, we collected two different types of plankton samples. We would do an 80 micron plankton sample and a 250 macrozooplankton sample. That 80 micron sample we would split up further, where we were looking at zooplankton and rotifers and phytoplankton. So for the ease of viewing, I've grouped all the species. So rather than give you individual species, other than the spiny water flea, of course, we're going to be looking at whole groups of things like cladocerans. And I'll have a list of everything that goes into each one. But it just makes the plots a little bit easier to look at. And going forward, before we had this data, before we were actually looking at our own trends of what's going on in Lake Winnipesaukee, we obviously looked to what does the previous research tell us for invaded lakes. And what a lot of the previous research said is that they document declines in Daphnia, which is a type of cladoceran. Some studies saw increases in diatoms, which is a type of phytoplankton. And other studies find declines in not only cladocerans, but also other groups of cladocerans and even cyclopoid copepods, which is a separate group of plankton. So all that is to say, I've been throwing a lot of names around of different types of plankton. You don't need to know that it's large-bodied zooplankton or large-bodied native zooplankton that our larval fish rely on. That's what has been documented as decreasing in other lakes that have been invaded by spiny water flea. So, a little unexpected these last few years. As I've mentioned, we had no detects of spiny water flea through 2022. We didn't find it until 2023 with extremely low densities of less than 1 per meters cubed. In 2024, we saw the average go up to nearly 4 per meters cubed, but a little surprisingly, we saw the average number of spiny water flea go down in 2025. So, and overall, these densities are very low compared to other lakes that have been invaded. For instance, Lake Ontario has densities of 20 per meters cubed to 140 per meters cubed recorded in their lake. Lake Winnipeg in Canada has reported 93.1 per meters cubed in the summer and 39.2 per meters cubed in the summer. So this is it. At this point, we do not have high densities compared to other lakes that have also been invaded by spiny water flea in North America. So you wonder, okay, these are low densities and we even went down in 2025. Is it having an impact at all? And unfortunately, it does appear that it is having at least some impact. So this is looking at our cladoceran density from 2017 to 2025. And this is a significant decline in total cladoceran density. So again, probably the most famous cladoceran that people may know is a Daphnia, right? They're the grazers of the pelagic area. They're a prize zooplankton for larval fish to eat. So that's probably the one that most people are familiar with. But the list on the right is the list of the different macrozooplankton, the different cladoceran that we have observed in these samples over time. And while I'm showing you the total cladoceran density plot, every single individual cladoceran species, or rather genus, that is highlighted in bold showed a significant decline over this time period. So we've seen declines in the Bosmina, the Cereodaphnia, Daphnia, Polypedium, all those in bold have declined. And despite spiny water flea being at a lower density in 2025, you can see that the density of cladocerans was lower than all previous years. And just as a little note to help keep things sorted, you might see in the upper right-hand corner that this is from the 250 micron mesh net. So because I have samples from 2 net sizes, if you're not sure what size net I'm talking about, just look in that upper right-hand corner, and that's the net that collected this data. So for cyclopoids, these are less targeted by larval fish. They're more of a native predatory zooplankton. And we did not see a significant decline over the whole sampling period. However, we did see a significant decline in one group. The cyclopoid copepods significantly declined, but when grouped with the calanoid copepods, that effect was washed out. Now we've moved on to results from the 80 micron net. And these are our rotifer densities. There's a lot of different types of rotifers you might find. However, no significant trends, and no significant trends by genus either for the list of rotifers that we found over these last few years. Now, cladoceran density in the 80 micron mesh net also showed a significant decline over this monitoring period. And once again, that significant decline is being seen across several genera of cladocerans. A little bit different result with the copepods in the 80 micron net. There was no significant trend over time and no significant trend by any group within the copepods. So the Nauplii are just the larval juvenile copepods. So it would be the babies of both of these together. You just can't tell them apart when they're that size. And phytoplankton from our 80 micron group, no significant trends by phytoplankton either. So, I was kind of curious for a little bit. We were starting to see almost a little uptick in cyanobacteria for a few years, but that really swung hard to a different group called golden brown algae this past year. So when you're doing statistics, there are no significant trends that shake out from here. And so I've got a lot of zooplankton data and phytoplankton data, but I think what a lot of people care about is the fish. So I can't speak to that quite as well as an employee from Fish and Game, but they were very kind to share their hydroacoustic forage fish survey using hydroacoustic pings on Lake Winnipesaukee. This is basically, pings that sense the density of forage fish in different parts of Lake Winnipesaukee, and the vast majority of those fish would be rainbow smelt. And I did find this plot very interesting because, as you can see, for every year that the Fish and Game conducted their hydroacoustic surveys, 2024 and 2025 are the worst years on record for their forage fish density. I think it's still a little too soon to say if that's something that will, will be an actual trend that holds, but it is a little concerning. The timing is interesting. So again, this is not my plot, but Fish and Game was very kind to share it with me for this presentation. So I've thrown a lot of plots at you. I've thrown a lot of information at you. What's the, general takeaway messages from all that information? Spiny water flea density was lower in 2025 than it was in 2024. That was surprising. However, even though the densities were lower, the impacts to cladoceran densities was maintained and even perhaps more extreme than the previous year. So we were seeing significant decrease in cladoceran densities in both our 250 micron samples and our 80 micron samples. There were no significant trends in the overall copepod, rotifer, or phytoplankton groups. As I mentioned earlier, there was a significant decrease in cyclopoid copepods in the 250 net, but that wasn't seen in the 80 micron net. And then when we're looking at least for forage fish densities, the forage fish densities were extremely low in 2024 and 2025. All right, so what are our next steps at DES? Well, obviously there's monitoring. We are going to continue monitoring our 9 sites, our 9 deep spot sites on Lake Winnipesaukee going forward. As mentioned earlier, this is something we do once a year. It does have to fit around other field work and obligations that we do. In fact, I'm not even in a biologist role anymore. I've moved into a data manager position, but this is a really important study, I feel, and one that I want to make sure continues. So we're at least hoping we're going to maintain that level of monitoring. However, we don't have the staff or funding to expand the plankton sampling at this point. They're very time-intensive samples to process. However, we are looking into a partnership with UNH to look into something called eDNA, which is environmental DNA. And this has been touted as an early detection system where you can basically take a scoop of water and see if there is spiny water flea DNA in that water, and it can flag that water body as potentially, having spiny water flea in it without actually collecting a physical sample. Obviously, that wouldn't be the the deciding factor of whether we thought spiny water flea was in a water body or not, but it could be a great way to help narrow down what lakes we should be looking at, just in case there were any positive hits out there. We're also looking to educate. So our big thing, especially with spiny water flea, but with other invasives as well, is clean, drain, dry. You'll hear it over and over again, clean, drain, dry. But this is the number one way to prevent spiny water flea from getting into new lakes, because once it's in a lake, you're not getting it back out. There's no treatment, there's no herbicide, there's no current method of removing spiny water flea once it's invaded a water body. Your best bet is to keep it out. And so cleaning your boat, draining all that water, hot water, like 140 degrees, has really been found to be effective at controlling spiny water flea and letting things dry out completely. We are also currently working on new signage to post at boat launches around the state. You can see the demo picture. It's not the final picture, but we are working on getting those signs printed so we can start putting them up at boat launches. And we also have a new tool for reporting any invasive species, obviously, used for spiny water flea, but really for anything with that QR code. It's a new survey from our exotic species program to make it easier for people to collect, to share what they're seeing out on these lakes, and if they have a concern that they've come across. And we're working on collaboration. So this past spring, there had a big meeting with nonprofits, the university, and the state, DES, and Fish and Game all got together in person to discuss the impacts of spiny water flea, to talk about prevention methods. We wanted to all kind of get on the same page when we were talking about sampling methodology and also those decontamination techniques with equipment. Newfound Lake Regional Association has been doing very frequent plankton samples, and they have already started the eDNA monitoring train. So that was actually, eDNA gave them a suspicion that they might have spiny in their lake, and it turned out to be true when they found actual individuals months later. So in that case, eDNA did work as an early detection tool. Newfound Lake, along with Lake Sunapee Protective Association, Squam Lake, Winnisquam Watershed Network, all these nonprofits have joined together to work to get by these Stop the Spread Swedish dishcloths, which they're planning on handing out to anglers and boaters this summer. They'll be stocked at local tackle and boat shops, and that template is available for any interested groups that want to make their own Swedish dishcloth. Yes. And so ideas for what you can do. I know I said it before, but I'm coming back to it, that clean, drain, dry. So New Hampshire Lakes has been maintaining a map of boat-friendly car washes around the state. They have the map on their website that you can go to and see when, after you're done boating and having a good day on the lake, you can make sure you can clean your boat down with hot water and give it a good scrub so that you're not spreading any nasty spiny water flea to any new locations. And they also have a mobile solar-powered waterless cleaning station that moves around as well. We ask that if you see strange suspected spiny water flea or any sort of invasive species, use that new tool to report it to DES. Usually people who are fishing are the first to encounter spiny water flea because it becomes entangled on their fishing line. It'll be clumped up on there. So if you see like anything like that while you're out fishing, something weird clump on your fishing line, take a picture, send it to DES. You can also get in contact with New Hampshire DES's new invasive species program coordinator. I have her contact information at the end of this presentation. Another option would be to become a lake host. You can share a lot of really important information with individuals who are coming and going and let them know about, the impact of spiny water flea and let them know that this is an invasive species that exists because not everyone is familiar that this is an invasive species and that it's in New Hampshire. And so with that, I'm happy to take any questions. My contact information is here on the page, and I also have the contact information of our new invasive species program coordinator. And with that, thank you.
Yes, thank you. So much information. We do have quite a few questions in here, so I'll start working through those. And Andrea, thank you for sharing some of those resources about our Lake Host program and the boat-friendly car wash stations. Those are in the chat if you want to take a look at those. Okay, so Donna's wondering, when will the spiny water flea signs be available?
They should be available this early summer. I believe they're going to get printed in May.
Great.
So yeah, in June or July.
And I think I saw a couple of questions similar to this one, wondering about water temperature. Are there, from other studies, are there water chemistry or water temperature thresholds for the survival of the spiny water flea?
Yeah, so they do prefer temperate, cooler lakes. So that's one reason why I mentioned that hot water on your gear, your fishing gear, or your boat is a great way to get rid of them because they do not like hot water. So they're very strong swimmers for plankton. They vertically migrate throughout the day. So during the day when it's hot and sunny, they do migrate down lower in the water column and will come back up when at night when they're ready to go hunting. So
Okay.
great question.
Um, and Janet is wondering how long can they survive on boat and fishing gear out of the water?
That's a great question. Um, so the problem with spiny water flea is they don't need a whole lot of water to stay alive. So if they're just like out and exposed with no water droplets around them, they're not going to last very long. That's going to be less than a day. The problem is, if they're on a fishing line and they're wound up tight in that fishing line, and there's little, basically a little droplet of water in there to help keep them hydrated, they can live for several days. So ideally, I know this isn't going to work for most people, but ideally, letting things dry out for 5 days is the best practice.
Okay. Greg is asking, how does the threat compare to the threat of other invasives and cyanobacteria? I'm not sure if there's like a correlation there or not.
Um, let's see. So spiny water flea do not have a direct human threat that we're aware of. So our big concern is the change in the plankton dynamics, the change in the energy flow, and its eventual impact on the fish community. With cyanobacteria, that has the potential to be more directly harmful to people and pets and livestock because it can, release toxins under some circumstances.
Right.
Um, as far as a relationship between spiny water flea and cyanobacteria, it's a little tenuous. There are some studies that have shown cyanobacteria increasing after spiny water flea invaded. There are other studies that have shown that cyanobacteria didn't really change very much after spiny water flea came in. It might be more of a lake by lake basis, and also there's other effects going on a lake, right? Like there's the temperature effects. There can be weather effects, whether new nutrients are being introduced into a lake that can have these bottom up influences on the cyanobacteria. So they're not really directly connected.
Okay. Bob is asking if you have any guesses as to why the density was lower in 2025.
That's a yes. I have questions. My initial impulse or my initial thought, which is not necessarily what's correct, but I'm wondering if they've already decreased the cladocerans and their prey species so much that they've already started to self-limit, which would seem pretty extreme because it's they're very low densities, but we also saw from those plots that the cladoceran numbers have bottomed out as well. So I'm not sure if they've already kind of eaten themselves out of house and home at this point.
Interesting. Okay.
But
Janet,
watch next year they'll like rebound in a crazy number and I'll be totally wrong about that. So who knows?
right. What is that? I heard a funny thing from the Forest Society about the years where there's a bunch of acorns and then the next year there's a bunch of squirrels.
Past
So
years, yeah.
yeah, yes,
Yes,
that's what it's
yeah.
called. Okay, so Janet is asking. have you seen the same type of decline in forage fish in other lakes?
Well, so DES doesn't collect that data. That is Fish and Game collecting the forage fish data. Um,
Okay.
I the Fish and Game did share forage fish plots from two other lakes, Sunapee and Newfound. Um, I I'm not going to remember. It wasn't this. It wasn't as drastic as that plot from Winnie. Sunapee, there I think was about the highest they'd ever recorded for forage fish this past year. Newfound, it may have been lower this year, but that would only be 1 year of overlap. So, um, and those are the only three plots that I've seen, and it's not a data set that I've worked with or am very familiar with.
Okay. Lee is wondering if the weather makes any difference for like a dry versus a wet summer.
Um, any difference in the spiny water flea population? I don't know. I think that's a great question. It's something I wondered myself when I saw those lower densities. And, we had a rather severe drought at the second half of the year last year. If there was any sort of link between that, I'm not sure. I don't have a good answer, but it's a great question.
Okay, let me go down here now and see. I think a few more have come in since we got started. Um, let's see. I'm not going to pronounce this right, but Craig Williamson, who's well known and will be at Lakes Congress this year, is asking Biothrepes. Is that right?
With the trapeze, yeah.
Okay. Produces resting eggs that can survive very harsh conditions. So, how effective are the available control efforts likely to be?
How, like the clean, drain, dry, is that what the question is?
I'm not,
I
maybe
believe that if
this
as
might have
long
been after
that
you
five
talked
day
about.
waiting period. Oh, sorry, go ahead.
No, no, please, yes. What?
My understanding is that the eggs, are not something that we're going to be hauling up with, your fishing line or your with the nets we're doing. It's possible you could be pulling up eggs with an anchor. So that's where, the dry period really comes into effect. So I think that's why, I mentioned earlier, 5 days is a long time to let things dry out, but that's best practice when it comes to spiny water flea to really make sure those things desiccate.
Okay. Rick is asking, are there any fish that feed on spiny water flea?
Great question. Yes, there are. Um, so when we talk about spiny water flea impacts to fish, we are not saying fish can't eat spiny water flea. They do all the time. The concern of why they impact fish populations is for the larval young of year fish. So when you think of a fish, they don't have hands and arms like we do. So as they're swimming around and they want to eat something, they can't just grab it and hold on to it and eat it. They have to use their mouths only. And so their mouths have to get around the entire prey item to be able to consume it. And the spiny water flea, like I said, they look tiny to us, but they're huge in the plankton world. So what we find is those larval fish, those tiny fish, are gape limited. Their mouths aren't big enough to get around the spiny water flea. So until they grow big enough to get their mouths around the spiny water flea, it's not a prey source for them. And that's where you start seeing that impact on the youngest generation. But there's evidence of different sunfishes eating them. Pelagic fish are, foraging fish that just consume plankton. Filter feeders will just inhale them in. There is some concern about impacts of the spiny water flea tail impacting their gut tract. So that big long spine with the hooks getting stuck in their gut tract. There have been some studies that say it doesn't affect them; they just pass the spines through. Other studies saying it can cause damage to the gut tract. So I think it's probably more of a species to species influence. But yes, they can eat them. It just kind of is a little bit more older fish can eat them.
Okay. Christine is asking if there's any research underway to figure out how to manage or eliminate them.
Not that I'm aware of. So I don't think there's any sort of treatment that, targets plankton, like individual plankton in any effective way, unfortunately.
Right. Janet's asking, is there a plan to set up plankton samples or eDNA testing at other lakes, or will that only be at the lakes that have sightings of spiny water fleas? Do you know that?
So the conversations that we were having with UNH, and this is still preliminary, we haven't finalized anything, but the idea would be to target like 20 lakes that we have, that are similar to Winnie in that they're, very big, large temperate lakes have, important fisheries on them. And just do a quick sample to see if we get a hit or not. That would be a one-time thing. And that list of lakes hasn't been figured out yet. We were hoping to consult with Fish and Game about, where they thought would be good places to look. But there's no plans to expand the plankton monitoring at this time, at least from DES. That's not to say that nonprofits won't be expanding their sampling efforts.
Okay. We'll see if anybody, oh, here, Rick is asking, where did they originate from? Was it Europe?
Europe and Asia, yep.
Yeah. And we still have about 15 minutes. So if you have any questions, those of you who are still on, please feel free to type those in and we'll get to them. I did have I was curious, and I'm sorry if I missed this somewhere along the way, but I've seen pictures of like individual ones, like you've shown several, but then we see them clumping on fishing line. And I'm wondering in their natural environment, like down at the bottom of the lake, are they kind of out swimming around or do they tend to clump up on plants and things? Like why are they like that on fishing lines so much?
Yeah, no, they're out in the environment, they're swimming around individually. The reason you're seeing them clump up on fishing line is because fishermen are trawling. So they're dragging that line through the water as, it's like going along behind their boat and it's going through a pretty long reach of water. And they just get hooked on that line and pulled because the line is moving through the water and they all just get bunched up there at the end. So that's not how they are in the water by themselves.
Okay. Betty's asking, is a once a year sampling enough?
Oh, probably not. Yes. So plankton certainly go through seasonal changes every year, and the densities will change throughout the year as well. To have a really good handle on your entire plankton community, it would be the more samples, the better. Would be the case. I think Newfound is currently doing every 2 weeks throughout the summer, so they're getting a really good. They're really capturing the changes that are happening throughout the summer and how their communities are moving and responding to the introduction of Spiny in their lake. Once a year is more of it's a very gross snapshot of what's going on. So that's why it's taken so many years to show even the most striking trend, and it probably will take many more years to show any other trend if it shows any at all. It's more of it's a monitoring effort where it was just more of a presence absence at that point. So they're ideally, if this was its own program, you would be doing a lot more monitoring for sure.
Yeah. Okay, well, here we go. Here's another one. Craig says the Pleasant Lake, New London Protective Association will be sampling plankton this year, both zooplankton and phytoplankton. There are places to send samples to be analyzed. Yeah, so I guess are there any like citizen science groups you recommend people reach out to if they want to help?
So I would encourage people, if they're not already involved in their own lake association, to get involved or reach out to their lake association. Lake associations are really good about collecting, not only water quality, but will also do plant surveys and plankton surveys and bathymetry and all kinds of things to help understand their lake better. So that would be my first stop. People who, if they're already involved in their lake association and they're wanting to collect plankton, working with their program coordinator, right? So there's a couple different volunteer monitoring programs. There's VLAP, for instance, and there's the Lay Lakes monitoring program out of UNH. And then obviously for the big lakes, there's nonprofits. They can reach out directly to the nonprofit for the lake that they are concerned about.
Absolutely. David's wondering what the life cycle is
Life cycle.
of the spiny water flea.
Yes. So I'm blanking on it all of a sudden. They will reproduce both sexually and asexually. So like a lot of plankton, they have asexual reproduction throughout the summer, and then once fall comes around and the conditions are starting to decline and they're going to be um, going into hibernation basically for the winter, that's when they will reproduce sexually, lay eggs that fall to the bottom. And then you don't typically, the reason that we started the monitoring program in late summer, that was the research and information I could find at the time, that was the best time of year to actually find them in your sampling efforts, so that if you weren't able to monitor on a frequent basis like we were not able to, that late summer was the time you could catch them. That's when they'd be the most abundant. Now, some more research that I have read more recently sometimes is showing higher abundances in the middle of the summer. So it seems to vary, like so many things.
Right, right. Bria, maybe you could chime in on this one. Janet's wondering if there are any pointers for lake hosts to better find the spiny water flea during an inspection.
Yes, that's a great question. So it's really just asking permission. So from the boat owner or the fisherman or whoever is going on the water or coming off the water to check those areas that are wet or could possibly be wet. So that's the anchor lines, that's their fishing line, maybe they have bait buckets. It's really getting that permission from that boater to look in those areas that might be sneakily wet.
Right. Okay. I think we talked about this a little bit, but what depth are they generally found, Kirsten?
Yeah, so they're not just staying in one spot. So like I mentioned earlier, they're strong swimmers and they can move up and down in the water column as they see fit. In the day, they tend to be lower down in the metalimnian and the hypolimnian, and they'll move up into the epilimnian as at in the evening. And so depending on the lake, the different layers of the lake, those different temperature layers, are going to be at different depths. So, but they can really be pretty much anywhere in the water column, not really at the top during the day.
Yeah.
So it might be safe
Okay.
to say, just on that, it might be safe to say, like during the day, if they're not going to be in that top layer, um, really working with people who are putting anchor lines down deep or even um, fishing lines, that's really who's going to see spiny water flea if the water body is infested.
Correct. Yes. It's almost always a fisherman who finds it first, it seems,
Mm.
because of the
Right.
fouling on their fishing line.
Yeah. So this is Daniel's asking if he's a lake monitor, how can he see them in the water? So I'm assuming it's probably pretty hard to see them in the water unless you're fishing and see them on the line.
That's correct. You're really not going to be looking into the water and seeing them swimming around. I mean, when you have an actual sample, like yes, they are, you can see them with the naked eye, but in the water, you're not going to see them unless they're clumped up on a fishing line, like you said.
As a, I have a kind of a question on top of that one. As someone who is a lake monitor, or if someone is out there like doing water quality sampling, could spiny water flea, if they're doing like a deep sample, could spiny water flea cling onto the line they're using to put down maybe like a Van Dorn or a water sampler?
Potentially, it could get snarled on that. Yes. Yeah. So that it's just that long spine with the two hooks, it gets caught on stuff. So if you're putting a line down really deep, it could get caught. It would be less likely than the lines where you're trawling and you're dragging it through the water for a really long distance. Less likely when you're in one spot, not moving and just down and up, but it's not out of the realm of possibility.
Andrea is sharing some photos there in the chat box of what it looks like when you see them clumped on the fishing line, and an image showing you the different layers of lake temperatures
Oh,
to go
thank you,
along with
Andrea.
our yeah for our conversations here, so you can see just how nasty those things look on that fishing line. Okay,
We
well, we do have a couple more minutes if anyone wants to put another question in the chat box, and then we'll finish up. Maria, did you have something else to add?
I was just gonna say that we just actually had our coordinator training for our Lake Hosts, for our Lake Host program, and one of the comments that came up about spiny water flea on the fishing line, it kind of looks like a pipe cleaner, like a gummy pipe cleaner.
Mm,
That's a great way to describe it.
yeah. Hey, well, we've had several people chiming in. Thank you for this presentation, Kirsten. So thank you again for joining us. I was happily surprised to see how many people registered. So clearly people are concerned about this new invasive species and are looking for some information on it. So thank you so much for joining us tonight. Now we'll have this presentation available for people to listen to. And thank you for sharing your slides. We'll put those up on the website as well. So if people want to go back and revisit some of that information, that'll be available to you. Um, and yeah, certainly reach out throughout the summer if you have additional questions, and we will do our best to get those answers to everybody. And thank you again for joining us tonight.
Well, and thank you for the opportunity to talk about it. So I'm really enthused to see how many people were interested in hearing about this topic. It's invasive species. It's a topic that's near and dear to my heart. So um,
Yes.
it's great to know that I'm not alone in that.
Absolutely, absolutely.
And thank you
Okay.
for all the hard work you guys do to train our lake hosts and get the information out there to the public and just really disseminate the information. It's a really really key important work that you guys do for New Hampshire lakes. So thank you for that.
Yeah, absolutely. And I actually, you just made me think, in addition to the interesting things that you were showing us tonight, the new signage and the new towel, New Hampshire Lakes, we did just produce a spiny water flea rack card, which has some detailed information on it about, those temperatures that we were talking about for cleaning your equipment and pressure washing and all that. So if you are joining us from a local lake association and you want to hand those out, or if you're a lake host and you want some extra handouts, please reach out to us and let us know, and we'll see what we have in stock and help get some additional information out to boaters over the summer.
Awesome.
Okay, well, great. Thank you so much. And I hope everyone has a wonderful evening. And oh, one last question, actually, since we do have a minute. Lee's asking, where can people find those signs when they're available?
Oh, that would be reaching out to the exotic species program coordinator. So Georgia Bunnell, her contact information is
Right.
right here on the page. So yeah,
Perfect.
she
Yeah, so
will be the one holding on to those signs.
excellent. Okay, well, great. Thank you so much, and I hope everyone has a great evening. Thanks again for joining us, Kirsten.
Yes,
Thank
thank
you all. I
you.
really appreciate it.
Bye.
Bye.